So our next speaker is Alba Gomez Segura, who is going to be talking on behalf of herself and her colleagues at the UK Centre for Ecology and Hydrology about their fantastic, innovative and Europe-leading work on image recognition and AI for species identification. So thank you, and over to you, Alba.
Thank you, Kieran. Yeah, so hi, I'm Alba, and I'm here as part of my group, which is led by Tom August and David Roy, and we are a group of 6 people, apart from me, sorry. And I just want to give a special recognition to Grace that we put this presentation together, but she couldn't be here today, so I will be doing the whole talk today. So for those that don't know, the UK Centre for Ecology and Hydrology is a research organization, and we focus on ecology, biodiversity, land and freshwater ecosystems, and their interactions with the atmosphere. And today in my presentation, I will be discussing several apps and projects that we have where we are trying to use AI to help us to identify species from images, and the collaborations we are building along the way to build the models to identify the species. So the UKCH has a long history of monitoring, measuring and observing the environment, and we are trying to support non-scientists or citizen scientists to collect data along with us. So here you can see a list of all the apps that we have available for free, and the idea is to gather as much data as we can, and it's normally carried from volunteers. They can be from experts or to occasional observers, and then we encourage them to go out, take images, and then try to help them to identify what is in the images. So the way most of those apps work is that they use the GPS and the cameras on your phone, and then people go out, can take a picture, and then the app helps them to identify different taxa. So for the rest of the first part of my talk, I will be focusing on those two apps. The first one is the iRecord, which is an app that makes easier to record wildlife sightseeings, and then the images that you put in the app are verified by experts. So then we use that data to support our science, and also to help decision making in local and national level. So before I dip into how we use this AI into the iRecord app, I want to introduce you the Nature Identification API, which is a computer vision model developed by the Naturalis Biodiversity Center, which is a center in the Netherlands. And 2 years ago, we were contacted by them to try to see if we were interested in to try their model in using the UK images. So this model was trained only in Netherlands species, but because it's close enough, we wanted to see the potential for using it for the UK. So we took the iRecords database and we filter all the images that were verified by experts, so that we knew that what was in the images, an expert said that's the true label for that image. And then we had almost 900,000 images that represented a bit more than 12,000 taxa all over the UK. And then the first results, and I just have to remind you, this was done on a model that had never been trained in UK species, only in the Netherlands ones. And we were quite impressed, actually. For some of the groups like dragonfly, moth, butterfly, birds, the model performed quite well. So here what you can see is the top one means the first. So the model gives you a list of 10 species and the probability of being those species. So in most of them, for these the top 5 groups, the model was sure on the top answer that it gives us with the higher probability on the species that it was. But then we also have the top 3, top 5, top 10, and none, which means the model couldn't know. And that means that the right answer was in the top 3, top 5, or top 10 answers. So we were quite impressed. So we make an agreement with them that we will give them the images from their records, and they will train a model for the UK species. So that's already in place, and it's implemented in the iRecord app. And the way it works now is that the people take an image, and then they upload it to the app, and then the app makes suggestions based on what the model thinks it is. And our idea behind that is to take advantage of AI to assist with identification. So the app gives you like several images of possibilities, so that people can compare if what they see is match any of the images that they are seeing with species and the probability. So so that it can help them to identify. But I have to say humans have always the last word. So if a human thinks the model is totally misleading, they can override the answer. But it's we think it can help a lot, especially people not that familiar with identifying certain species, to encourage them to go out and try to identify them. Also, the idea behind this is maybe to help experts with the overload of images that they need to verify. So maybe we cannot trust the AI to identify everything to completely trust it, but maybe we can use it just for the images that is always very, or the species that is always very confident, we can maybe leave those as the AI, but then the experts can focus mainly on the ones that are more difficult to identify or are more rare, or the model just doesn't perform well on them. So we think it's a combination between still the experts, but the AI can just help to reduce the number of images that they have to identify. Because as I was saying, the model still has a lot of limitations. So I just wanted to show you here an example of the Adonis ladybird. So in the first image on the left, there is like the model is almost 100% sure that to the correct ladybird. But in the second image, that for us, it could look like it's clearly the same thing. It actually dropped to 33% sure. So it's very low, which, yeah, like you cannot really tell that the model knows what is seen. And then in the last image, which is still the same species, it totally confuses with another ladybird. So clearly the model, it could be different things. It could be that all the training images that the model had were always on the same background with the same light. So it's not just the features of the actual insect that is looking, but also the surroundings of it. So that means that the more images and the more different backgrounds and locations that you can give, the better the model will perform, and more generic it will be. So if you always have the same background at the back and then you give it another one, it might perform very low. So the second app I wanted to talk about is the E-surveyor app, and this app is a part of a project which is called Ag0 Plus, and the ambition of this project, which is a 5 years long project, and is helping the UK transition towards domestic food production that is sustainable, carbon neutral, and have a positive impact in nature. And this app, what it's doing is helping farmers to assess the quality of the habitats that they manage, so that the idea is the farmer goes on the age of the crops that they have, and they take pictures of the wildflowers of any plant that they have around a crop land, and then the app helps them to identify the plant. And then we use like data that we have been collecting for the last 10 years to give back a report saying which insects are supported by that plant, so which pollinators will fit in that plant, also which natural pest controls insects will fit on that plant, so that they can assess the quality of their land in terms of pollinators. So future opportunities or things that we think that this has a lot of potential. So we think it has a lot of potential to help people to identify. Still, there is a limitations and experts are always needed, but we think the AI can be used, like maybe if we combine it with other data like phenology, time of the year, location, maybe we can help the models to perform a bit better. Also, like the app can help to flag to experts like misleading identifications that maybe help them to filter which images or which species to focus first when they are verifying the images. But those apps are there. If you ever use them or you have any feedback, please let us know, because it's always great to know what people think about the implementation. So this was the first part of my talk, which was a more generic one. And the second part, I'm going to focus on the AMI system, which is an example of a tool that utilizes computer vision and cameras for identifying insects, in this case, moth. And I'm going to especially show you examples on how is this system performing in the UK moth species. So AMI stands for Autonomous Monitoring of Insects, in case you didn't know, and it offers a platform for long-term, non-lethal, time-efficient, and standardized monitoring of insects. So we think about it that is a good alternative to manual collection, sorting, and classification of the insects, and can help a lot to save time when you are analyzing. So why moth? I guess that's one of the first questions. The first thing is moths are very important pollinators, like they are a food resource to many species, and they respond very quick to changes in the environment, so that makes them a very good biodiversity indicators. Also, it's very easy to attract them, so it's well known that moths are attracted to light, so it's very easy to attract them and take pictures of them. And also we think they are different enough, there are enough differences between species that a computer vision model can perform okay on them. So this is the how the AMI looks like, and this is the first prototype. It was designed by and researchers at the University of Aarhus in Denmark. And the first prototype had a UV light and then a whiteboard where the moth land, and then it had a camera and a Raspberry Pi, which is a small computer that was controlling the camera, and then it was saving the images to an SD card. And the way it works is every time the camera was detecting motion, it was taking an image every two seconds for 2 minutes, and if motion is still going, it will keep going. But then after that first websites come along in 2021, the CH researchers took it and brought it to our workshop, where our engineers try to make the model more resilience resistant and more scalable, so that we could build lots of them and put them around the UK at the beginning and then around the world in other projects. And also we changed the light. We are using a LEPI light, which is a combination of a green, blue UV light and a white light that it's supposed to attract more moths. And we are still using the same camera and the concept of the Raspberry Pi and saving the image into an SSD. And I have to mention that the system is powered by solar panels, so it's totally independent and in terms of energy, if you put it in a place where solar panel can work. But there is a possibility also to plug it to mains if solar is not a possibility. And so as you can imagine, the quantity of images that this device can take is crazy. I remember one project that we had that we bring one to Panama. There was one night that we ran it for 10 hours, and the next day we had 20 gigas of data. And it's just because there is so many biodiversity and so many insects around there that it was just a video that we had an image for every 2 seconds for the 10 hours. So after a few experiments in other hotspots, biodiversity, we decided to move away from motion, and we are doing snapshots now. So we take an image every certain amount of time. And the rule we are following is it depends where we are. So in places we have this big project that is trying to bring the AMIs to hotspot biodiversity countries. So we have in Costa Rica, Panama, in Kenya, in Singapore, Thailand, and in those places we are taking an image every 10 seconds just because the biodiversity in these places is very high. But in places in the UK, where we have less activity and less number of species and they are very well known, we are taking an image every 60 seconds or every minute or every 10 minutes, even in some places. So here you can see the AMI in action. That video was taken by Jenna Lawson in Costa Rica last February. So you can see how it, the light is on and how the moth land on the board, just to give you a bit of context. And then, so how is the AI applied in the AMI? And for that, we partnership with the Mila Institute, which is a artificial intelligence institute in Montreal, in Canada. And there are 4 models that we run in the images when we collect them. So the first one is a localization model, and this model, what it's trying to do is find all the insects in an image, all the insects. It doesn't matter if they are moths or not, anything, all the objects in an image. Then we have a second model, which is a binary classifier, and in this we take all the objects that they were found in the first model, and then this new model tries to classify them to moth or non-moth, so to just reduce the number of specimens that we need to classify to moth species. We are not throwing away the non-moth insects. We're still recording them. It's just that we don't classify them to the species level at the moment, but we think in the future this system can be used to identify other taxa. So we think it's good not to save the other insects. So as I was saying, the third model is the classification one, where we take the ones that were classified as moth and we try to put a species level on them. And finally, we have a tracking model, and this one is trying to see if we can track when an insect comes for the first time in front of the camera, if we can track how it moves around the whiteboard, also how long they stay, at what time of the night they come in. And of course, this has some limitation, like counting insects is still not possible because overcounting is very difficult, because if a moth leaves and then the same moth come back, we will counting at twice. But we think it can help a little bit with activity, and in the future, it might be possible that we can do more stuff. So in this video, I just wanted to show you the ML models in action. So here you can see some of the raw models or the raw images from Panama. And then we do the tracking and the object detection, and you can see how we can track where the moths are going. And then the moth classification is done on UK species, and you can see the blue are the ones considered moth and which moth they are, and the gray ones are the non-moth ones, and the probability of being a non-moth. This is that. So, what tools there are available to then analyze the data and run those models on the images? And for that, we are working with North American and other countries in Europe, like Denmark, to build a processing tool. And one thing that is already available is a GUI, which is a graphical user interface that you can download on your computer, and then you can run the model of your country on your images. There is a bit of limitations on that because most laptops doesn't have a GPU or capacity to maybe run the whole night of images, but maybe you can just run on a subset and see how well the models run on your images. But the idea is at some point this year to have a website interface that has more capabilities and people can actually run the models on all the images that they have. So here is some screenshots of how the website is going to look. So you will have a page for projects where you can just create the project for your AMIs, and then you will be able to run the models and see which species are there and see some information of the species. There will be some statistical analysis done on your images for you, but also there will be like the metadata, like where are your AMIs located, and extra information like the time, when is the sunset and sunrise in the countries that you are running the AMIs, and extra information like that. So now I wanted to talk a little bit about the model and what I mean by accuracy and how are the model trained, so that people have better understanding and what is going on behind all those models. So for training the UK model, we use a dataset that had more than 2,500 species, and it was covering both micro and macro moths. So I think we had around 900 macro, and the rest were micro moths. And then, as a general rule, so all these models are trained on GBIF images, because when we started, there was no data coming from the system. So we thought GBIF was a good place to start, to have a data set that we could start doing something. So the rule was like querying GBIF on those species and get a maximum of 550 images per species. And if there was less, it's less. But we required a minimum of 6 images per species and a maximum of 550 images per species. And this is because when we split the data set into training and test images, we need at least 5 images for, so we did it in a ratio of 5:1, so at least we need 5 images for training and 1 for testing. And when I mean training and test images, is like the training images is the ones that you will use for your model to learn about the species on them, to try to extract characteristics of features of your species. And then the test is when you have your model trained, you run it on your test images, which are images that it has never seen before, and then it will try to classify them. So it's a way to test the accuracy and the performance of your model. So if we take this example of the elephant hawk moth, we have 6 images and we split them in the training and the data set. So once we have the model trained on those 5 images, we just test it with our test image in that case. And here, for simplicity, we will have a score of 100% if it's decided that is the elephant hawk moth, but we will have an accuracy of 0% if it's not, just because we only have 1. But in the real experiment, we actually had 550 images for this moth, and then we had out of those so we had 600 images for training and 150 images for testing. And out of those, the model performed, like it tells us the correct moth for 146 of the images. So it means that it has an accuracy of the 97% for that particular moth. And we can do that for all the species in the UK. And here you can see the number of species for 10 of our species, and how many images were for training, how many images we used at the test, and the accuracy. So out of of those test images, what was the accuracy of the model. And there is a general pattern that shows that the more training images you have, the higher is the score. So you can see the species with the maximum number of images, like the first one, they can have up to 99%, 91% of the model always being right. But there is also, on the contrary, for those we have very few images. So the last one that we just had the minimum, the accuracy is always very low. It's just the model didn't learn enough to ever give that name to any of the mob that it have seen. So we just wanted to highlight that the importance of how many images you have and how variable your images are. So also I have to mention that the accuracy varies between species. Like for 1/3 of the species, we have an accuracy of 90% of like an accuracy of over 90%, but for more than 60%, we have a 70% accuracy. So it's not too bad, but it's not just the number of images that influence the accuracy of your model. It could be also like the quality of the images, the specifics of that species. So there is different things to get in account apart from knowing how many images that you have. And for that, I just wanted to show you with a more visual example. So in this one, all the species in that slide have the maximum number of images. They all had 150 images, but not all of them performed very well. So in the top row, you can see that the model performed quite well for all of them. And this could be for several reasons. It could be that the characteristics of the species are very unique, like the colors, the pattern, the size is very unique. So it's very difficult to confuse them with another one. But if we look at the ones in the second row, which still have a lot of training images, the first one, for example, the light male yellow underwing, it's always a 5%, which is very low. And it could be because it's very similar to another species, so the model couldn't tell them apart. So it's always giving us the wrong name. Also, the second one, which only 33%, it could be that the moth doesn't have any distinguished pattern. The colors are very light. The wing colors just doesn't make anything particularly particularly interesting for the model to learn. So it just never picks up anything. And then we have the case of the third one, the dark amber, that it has a multivariance. So it depends on if female or male. The colors are different, and they can be more dark, more light. So it could be that the model also doesn't know which features to learn about that species, and that makes it very confused. So there is several things to take in account when you assess the accuracy of your model. And then here are some examples of species that are quite distinguished, but because we have very few number of images for those three, we only have 6 images. So we have 5 for training, 1 for testing. So the model actually performs 0% for all of them, even though they are quite distinguished. Unless we manage to increase the data set, the model cannot pick up enough information from the images to be able to identify these images correctly. And then I just wanted to show you how many images we need are sufficient to train a model, and when we can stop collecting images. And Grace did that. He did like linear regression on the images and put this plot where it's showing you the number of training images of the accuracy of the model. So the blue line represents the overall trend, so indicating that in addition of more images, we see an increase of the performance of the model to a certain point where then it goes a bit flat. And then the line, the red line, the one that is dotted, is just showing you beyond which point the images don't really add any extra features. So the model performs the same if you give them that number of images or more. So we can see that the point is around 200 images. But to be honest, if we look at the graph, around 100 images is enough to have enough information to train a model. And this, of course, can vary between the species, but this gave us an idea of in which species we might need to focus a bit more on getting more data, and which ones we already have enough and we just don't need to collect any more data. So another test that we did is we wanted to try to see if we could compare the model performance to human identifying the same insects. So if we can see if the model struggles with the same species or the model struggles with different species to the human eye, because they don't need to follow what we follow visually. So we just wanted to compare if there was any comparisons between what the model learns from an individual and what we humans look at. So for doing that, we use a data set which describes how difficult it is to identify a moth, so by experts. And then 1 means it's a very easy to identify, 4 it's being impossible to identify only by looking at an image, and indeed you need to maybe dissect the individual to be able to tell which one it is. So here we can see for a 1, the herald, which is a very unique shape moth, which is very easy to identify, but the 3 species on the bottom, they are quite hard to identify only from images because they look very similar. So when we did that experiments, like we split the moth in the list from these 2 groups, and then we plotted against the accuracy of the model, and we find that actually the species rated as difficult by taxonomic experts was also the ones that the model struggled more to identify. But even that was the case, there is still surprise that the model achieved over 50% accuracy on for a lot of the species which are difficult rating. So it might indicate that in some cases where it might be clear for the model which species it is, and they can tell them apart, maybe looking at the pixel level or something like that, where for humans it might be more difficult. But it seems it follows the same trend, like the ones that are very easy to identify by humans, the model finds them easy as well. So as a conclusion, as a wrap up, so I talked about the AMI and how it can complement traditional monitoring by allowing us like having this long-term continuous monitoring and fast results, and the collecting a lot of data continuously. We can put this system out and they can in remote reserve, and they can run for months without no human interaction, apart from going and collecting the images maybe once or twice a month. And then but maybe we think that in places where we think these systems might perform the better or might help us the best is on those places which maybe biodiversity of moth is very understudied. So in places like the tropics, where there is still a lot of unknown species, we think this system can help us to collect data and start having samples of images of species that never have been described before. But also there is some potential limitations and next steps that we are looking into. So the first thing, and very important, is what is the threshold of confidence? Like for some species, it could be that if the model is 60% sure, we can trust it. But from all the species, it might be that we need to be at least 80 or 90% to trust the model. So this is something that we still need to work with taxonomists to get into a number, if not more generic, maybe in group of species. So that means that we need label data. So we are already working with experts, especially on local countries like Costa Rica and Panama, that they will be labeling the data coming from the system to help us to build a better data set. And so despite all the advances of machine learning and the potential of these tools, we still think it's crucial to work with experts on identifying the insects. We just think this can help us to move forward in the field. And also, like, some of the next steps that we are working on is exploring the addition of acoustics monitoring for birds and bats. So we already have 20 systems this season in the UK that have incorporated a microphone for recording those 2 groups, and so that we hope to increase the capabilities of the system. And also we have a project with the Turing Institute in London to try to build edge models. So an edge model is a model that will run on the system. So now we need to go collect the images, download them, push them to the cloud and process them. With these edge models, we could start doing some of the processing inside of the Raspberry Pi, inside of the computer of the system. So we hope that will help also to be able to analyze the data more quick. But we think the system has a lot of potential, and in the future years, the models will just go better and better. So we really are looking forward to see how it goes. So that's all for me. Thank you so much. And if you have any questions, please let me know.